Welcome back to Polymeric Biomaterials. We are starting off on a new theme from this lecture onwards, namely surfaces and interactions. So far in the course, we have largely focused on the bulk properties, how to think about processing and how that affects properties such as porosity, pore size, microstructure and so on, which are all extremely important for all biomedical and clinical applications. But equally important, if not more, are surfaces and surface properties. So that's sort of the theme that we're going to focus on in today's lecture and in the next couple of lectures. So with that background in mind, we're going to be today looking at some general comments on biomaterial surfaces, the role of ECM interactions which can form the interface, as well as some aspects related to surface modification from an engineering perspective. So in today's lecture, we will first start off by looking at what surfaces mean in general. We'll look at some definitions, and we will also pay some attention to why surfaces are different from the bulk, how do they differ, and very importantly, we will also focus on the role of surfaces and surface properties in promoting or inhibiting specific kinds of interactions. Now, we will later on in this course look at many other types of surface-driven biological processes, starting from protein adsorption to blood compatibility to immune response, inflammation and so on, and all of which will be determined largely by surface-based interactions. We will next look at ECM or the extracellular matrix and the interactions that they can promote. So we will look at the components of the ECM, many of which we have already looked at under the topic of natural polymers already. We will then take one specific example of cell adhesion and try and look at how specific ECM proteins along with transmembrane proteins such as integrins can promote cell adhesion. And this is just for us to illustrate why surface mediated interactions are important and how biomaterial surfaces can be specifically designed to promote specific kinds of biological responses. And towards the end of this lecture, we will look at modification of surfaces. Essentially, we'll broadly look at physical, chemical and biological methods to modify surfaces. And we will look at one example each of them, namely plasma, silanization and a very famous covalent coupling chemistry called the EDC-NHS chemistry. So with that quick overview of today's lecture, let's jump right into the lecture. So before we begin looking at what it means for surface properties to have an effect on biological responses, we will first offer some comments on the surface itself. And to contextualize it, we're looking at a quote from Poly, which sort of very interesting, because he says, "God made the bulk and the surface was invented by the devil." Now, this is a tongue-in-cheek comment, essentially to convey the complexity involved in studying and controlling surfaces. Surfaces are intrinsically quite different from the bulk. Surface properties are very different typically from the bulk, and controlling surface properties are also very difficult. So one way for us to appreciate surfaces is to first ask what is meant by the surface, right? So we first define what we call a surface, and we call it the zone where the structure and composition influenced by the interface differs from the bulk composition and structure. So if you think about this from a biomaterials perspective, you have a biomaterial surface. This part here is the surface. This is all the bulk, right? And if this is placed in a biological host where it will be exposed to an aqueous buffer, there is the buffer bulk here. So this is the bulk for the material. This is the bulk as far as the host is concerned, essentially the buffer. Now, this particular layer here, a few layers here of the biomaterial right at the interface is quite different because it's different both from this bulk here as well as this bulk here, and is affected by the interactions between the two phases. So that's what we are saying here, influenced by the interface, right? And one way for us to appreciate why surfaces differ so much from the bulk are, the first thing is surfaces are highly reactive, unlike the bulk. This is particularly true for polymers. The second is surfaces also show mobility and can change their properties with time. Third, and for that reason, they can also be unstable, and they also contaminate very easily. So you notice here, if you look at surfaces from this perspective, you'll see that these properties are generally not true for the bulk. Although, for example, the bulk may show changes over a long period of time due to aging effects, those effects can occur over a much shorter time scale for surfaces, right? So even some of the effects that can show up on both in the bulk and in the surface have very different manifestations when it comes to surface versus bulk. And so this set of properties makes surfaces very, very difficult to control and study, right? That's sort of why perhaps Polly said the surface was invented by the devil. And from a biological perspective, what we note here is that the surface is the only portion in contact with the physiological environment. So the host or the cells or the buffer that the biomaterial will eventually be sitting in will first see only the surface of the biomaterial, right? So that's what we say here. Host reads the surface and responds to the chemistry and the structure present at the surface. Now, there could be long-term repercussions occurring due to bulk properties, but almost immediately the first thing that the host will notice through adsorbed proteins is the surface itself. And one shouldn't forget that the host has an aqueous buffer at 37 degrees Celsius, and so whether we like it or not, as soon as the biomaterial is implanted, the surface is going to be hydrated, right, depending on what type of a surface it is, where ions can also come in and interact. So it's a very complex set of reactions or processes that happen as soon as the biomaterial comes in contact with the host. And it's dynamic. It continuously keeps changing. That's why controlling surface properties is also complicated. And because of its importance in its interactions with the physiological microenvironment, surfaces primarily drive biological reactions and responses occurring in response to the biomaterial. And this could include anything from protein or cell adhesion to blood interactions to inflammation to immune response, all of which we will look at in great detail in subsequent lectures. From a material perspective, the properties of interest typically for surfaces include surface roughness, surface wettability, the chemical composition of the surface, stiffness, charge, and morphology. And there are very standard methods for assessing and characterizing surface properties. For example, the morphology can be assessed using electron microscopy, for example, scanning electron microscopy. You can also have wettability assessed using contact angle measurements. These are very standard sets of measurements that are made to characterize the surface properties of biomaterials. Contact angle also is a measure of surface energy. The chemical composition can be assessed depending on what your interest is, using a bunch of different characterization methods. For example, XPS is a very common method to look at the first maybe few 10 nanometers maybe or so. If you want to go a little bit deeper, you can look at something like FTIR, but in the ATR mode, which can go up to 5 microns. That's no longer considered the surface per se. You can also look at stiffness, which can be assessed using atomic force microscopy. You can generate force displacement curves. So these are all standard, very, very commonly used techniques to assess the properties of the surface. And one important thing to keep in mind is these properties that you get from the surface will typically be quite different from what you will get in the bulk. one example of that is stiffness. So if you make a scaffold using one of the techniques that we have seen in earlier lectures, for example, electrospinning, you will get a very porous scaffold. So you can do tensile testing, bulk mechanical tensile testing on it, which is again something we have seen earlier, and you will be able to get a specific modulus for the material, right, in the bulk. But if you take the same material and in the electrospun form and you try and assess the properties of the surface, the stiffness of individual fibers using something like AFM, you will get very different properties, right? So surface stiffness can often be quite different from bulk stiffness or bulk properties. Now, they are both important. Bulk properties are more important from a macro performance perspective in the long term, and surface properties such as surface stiffness, for example, are quite important when it comes to biological interactions. For example, when cells attach to a material, what they are feeling is not the bulk modulus or the bulk stiffness, they actually sense the local stiffness, right? And so this is a very big area called mechanobiology, where people study and try to understand how cells respond to differences in local microenvironments, particularly differences in mechanical properties locally. But our goal here is not to get into the details of each of these characterization techniques. There are other standard textbooks that you can refer to if you want to get into the details of the principles of each of these techniques. Or if you are working on some of them, you will already be perhaps familiar with many of them. What we want to point here is that these properties are particularly important for us to assess when we prepare biomaterials for specific applications. And these are some techniques that you can use to characterize these properties. If you ask how far does or how deep does the surface extend in a biomaterial, the answer, it turns out, is not a straightforward answer. One can talk about a length scale, right? So for polymers, typically you're looking at tens of hundreds of nanometers, right? So that's sort of what we are saying here. 10 to 100 nanometers is a reasonably good estimate for the length scale of the surface itself. Again, it depends on the polymeric system, the type of processing you've used, and the molar mass. Now, so that was all from the material perspective. Now, from the host perspective, you can ask the same question. Essentially, what is the importance of surface properties in promoting specific kinds of biological interactions? And so that's what we're looking at here. As we've already hinted at, surfaces play a very important role in dictating a whole host of processes, right from hydration to protein absorption, to platelet adhesion and activation, to neutrophil responses, macrophage responses, inflammation, everything else that happens thereafter is largely governed by the surface, and all of which we will look at in subsequent lectures in very great detail. But the point here is to drive home the importance of surface properties for us to essentially appreciate the importance of surfaces in biological interactions. So surfaces play a very important role in determining biocompatibility. Everything I have said are different dimensions to biocompatibility, right? Whatever I've said so far in terms of biological interactions are all different manifestations or different dimensions to biocompatibility. Surfaces also play a very important role in performance and failure, because if you, as we will later on see, if you don't have the right kind of surface properties, you can end up with essentially a very, very thick fibrous capsule that encapsulates the entire device, and that's going to lead to eventual failure of the device. And like we have said, surfaces play a very important role in promoting initial biological responses, some of which I've already mentioned. They also mediate specific cell interactions or cell material interactions. They can also inhibit, as we have seen earlier or hinted at, where we've looked at the idea of non-fouling surfaces. We've talked about peg chains being attached. That's again a surface-driven phenomenon, right? So you're not changing the bulk, you're taking a surface and grafting peg chains onto it in order to hinder or retard protein absorption and cell adhesion. And of course, you can have a very important role for surfaces in mediating degradation and aging effects. For example, if degradation proceeds through ester hydrolysis, water still has to penetrate from the exterior, from the surface inwards, right? And for water to have access to the inner regions of the bulk of the polymer, we have to first hydrate the surface layer and then slowly proceed, right? We have seen some aspects of this earlier. Of course, aging effects also are very critical when it comes to surfaces. Now, one quick example we will look at for why surfaces can be very important in a clinical context is surface structure playing a very important role. So, we are looking at the example of polyhema, which is a very famous polymer in biomedical applications, particularly in its use in contact lenses. So, shown here is the structure, and right away you notice that there is a methyl group that sticks out here, and there is an OH group that sticks out here, and those two have very different properties. One of them is not polar, the other is polar, and it turns out that that difference can have consequences on which groups get exposed on the surface depending on which microenvironment the lens is placed in. For example, in air, all the methyl groups will stick out, all the non-polar groups will stick out, and all the more polar groups will stick inside, right, if you just look at the surface. And this is again to minimize the surface energy. But if you now stick the same thing in an aqueous medium, in water, which is typically where these lenses are employed, now you see a reversal. These groups can sort of rearrange, and you can now have the polar hydroxyl group sticking out, because them sticking out now helps them favorably interact with water, which again overall lowers the free energy. So again, all of this is driven by thermodynamic considerations, essentially minimization of free energy. In the former case, the methyl groups sticking out in air, the non-polar group sticking out in air is more energetically favorable, while in water or in the aqueous medium, the hydroxyl group sticking out becomes more energetically favorable, and there is experimental evidence to show that this rearrangement happens. So, if you recall a moment ago I said the surface is highly mobile, this is what I meant. So, this is one manifestation of mobility at the surface, and these hydroxyl group sticking out in the water is particularly important for hydration, for tear film stabilization, and so on. So, it makes sure that the lens performs the way it is supposed to perform. So, again, we are looking at a structure of the material itself directly affecting the properties and function. So, what we want to do next is take a slight detour and look at what I am calling the interface, right? And this is for us to better appreciate later on why surface properties are very important for biological interactions. And we will specifically now focus on the extracellular matrix or the ECM. Right. So, this is the matrix that adherent cells secrete to attach to biomaterial surfaces, and those attachments are mediated through proteins, right. So, because the ECM forms the sort of the bridge in some sense between the cell and the biomaterial surface, it is very important for us to study and understand the components of the ECM and the functions and roles of the ECM so that we can better appreciate how cells can respond to specific kinds of surface properties. And it will also give us clues as to how we can better design materials with specific kinds of surface properties, chemistries for example, to promote specific kinds of biological responses. Now, the ECM itself is not new to us. We've already seen many of the components of ECM in earlier lectures on natural polymers. So, the primary components of ECM largely include fibrillar and structural proteins such as collagen and elastin, which we have looked at in great detail. Specialized adhesive proteins such as fibronectin and laminin, some of which we will look at in this lecture. And amorphous macromolecules, namely proteoglycans and GAGs, essentially charged molecules that can interact with water extensively. So, this is also something we have seen in earlier lectures. And of course, the ECM also has water and soluble cytokines and growth factors. Remember that the ECM is not just playing a role in mechanical support, it also serves as a storehouse of many signaling molecules, right. So, it's one way through which the cells can interact with their microenvironment, whether it is the biomaterial surface or with the other cells in that tissue. So, the primary functions, like I just said, include mechanical support for cell attachment, that's obviously one of the most important functions. ECMs can also direct and dictate cell orientation, growth, and migration. And you can take advantage of some of these things when you're preparing, let's say, biomimetic surfaces. You can prepare surfaces or materials that have specific kinds of orientation or alignment in the biomaterial surface, such that when cells start to deposit ECM, they will also be aligned in that direction, right? One example of that is aligned nanofibers prepared using electrospinning. They establish a functional microenvironment for the cells to function. And like I said, they serve as a storehouse of signaling and regulatory molecules. And this would be a good time to pause and recall the structure property relationships that we have seen for collagen and elastin in lecture 19, as well as proteoglycans, GAGs, and hyaluronic acid in lecture 20, all as part of natural biomedical polymers. Now, the basal lamina, just as one specific example, is a specialized type of ECM, which is about hundreds of nanometer thick. It's sort of a 2D mat in some sense that underlies all epithelial sheets, right? So it's a specialized kind of ECM, very thin, and that's why we're looking at it in the context of surfaces. And it's composed primarily of type 4 collagen and laminin. The basal lamina acts as a selective barrier in specific kinds of tissues and also plays a very, very critical role in wound healing. Now, from a design perspective, if you want to create biomimetic surfaces, one of the strategies that people can use is to use collagen and laminin, right? So you can prepare materials that are now surface functionalized with collagen and laminin. Now, many of the natural biomaterials, as we have seen earlier, have very, very good bioactive sites for and motifs for cell recognition, but many of them don't perform very well when it comes to bulk mechanical properties. So one can take advantage of both natural and synthetic biomaterials by preparing bulk materials from these synthetic polymers that have very good mechanical properties and performance, but also surface functionalizing them with molecules such as collagen and laminin that can promote specific kinds of biological interactions. So that's one very commonly used strategy from a design perspective. And again, just to drive home the point about the role of the ECM being very important, we have already looked at the structure property function relationships in articular cartilage, specifically with regard to role of the collagen and proteoglycans. So this was something we looked at as part of viscoelasticity and articular cartilage in lecture 25, essentially where we saw that the charge on the GAGs in the proteoglycans help interact with ions in water to achieve internal tissue pressurization in the cartilage, which allows very high compressive loads and a microenvironment with a balanced hydrostatic or osmotic pressure to regulate chondrocyte function. So the chondrocytes are the primary cell types found in articular cartilage, and it almost acts like a sponge in some sense, the cartilage. So when you stand up, you put compressive loads in the cartilage and the water pushes out, goes out of the tissue, and when you relax, the water rushes back in because of the negative charge, highly negatively charged molecules present in the ECM of the articular cartilage. So this one tissue serves as a good example of how collagen and proteoglycans together regulate the mechanical and biological function of specific kinds of tissues. One more important point to keep in mind when it comes to thinking about ECM is you have to also contextualize some of these properties that we looked at to specific tissues. So one way for you to understand or better appreciate some of these functions is to now ask, what does the ECM look like, for example, in a tendon or a ligament as opposed to a bone, right? In a tendon or ligament, we have already hinted at the role of aligned collagen fibers, crimped collagen fibers, when we looked at mechanical testing in lecture 24, playing a very important role in having that triphasic tensile kind of a behavior, right, where the toe region is there, followed by the linear region and so on, right? All of those can be mapped back to the changes that occur in the structure of the collagen, which is the part of the ECM of tendons and ligaments. Likewise for bone, you will have not very aligned collagen fibers, but you will have mineralized collagen fibers, right? You will have hydroxyapatite like mineral present, and that strengthens the tissue through the ECM, right? So again, just two quick examples of two different types of ECM for you to better appreciate how ECM regulates cell function. For example, in bone, the ECM can serve as a storehouse for BMP2, which is a very important protein for bone growth, right? So again, just to help us appreciate the functions of ECM in specific tissues. Now, moving on to looking at one particular example of a biological response. We've hinted at biological responses throughout, but if we dig a little bit deeper and look at one specific aspect of the ECM, namely fibronectin, and then ask how does it play a role in promoting cell attachment. So this is just one illustrative example. We will not be looking at all kinds of cell responses. Cell adhesion is just one type of a cell response. There are many others such as proliferation, migration, differentiation, polarization, lots of them. We are just looking at cell adhesion as an example from a biomaterial perspective because that's the focus of this course. So let's first look at fibronectin in more detail. It's an ECM protein, as we've already mentioned, that aids cell adhesion. Now, the cell adhesion is not aided by the entire protein itself. It's aided by a specific tripeptide sequence called RGD. And by now, if you've paid careful attention to the lectures on proteins, you will be able to figure out what R, G, and D stand for. They are the single letter abbreviations for different amino acids. So I would encourage you to go and look up what R, G, and D stand for. So it turns out that this specific tripeptide sequence is very important for recognition by cells. And the recognition is done by integrins, which are transmembrane proteins found in cells. So it turns out that on its own, the fibronectin is present in the soluble inactive form, and it requires specific binding or association with receptors such as an integrin to be able to form fibers and thus promote stable attachment. And they, needless to add, they promote adhesion, proliferation, and they're also implicated in wound healing and a number of other responses. Now, we've already hinted at the fact that fibronectin has to, or the RGD sequence particularly in the fibronectin has to attach to specific integrins found on cell membranes. Now, shown here is a cartoon of that picture, that binding, where this is the cell membrane in itself. What you have here is the ECM, right, these fibers, and shown here is that specific RGD sequence which is found on the fibronectin, right. So you only focus on that RGD for now, and then we see that the RGD is now able to bind to integrin, which is a transmembrane heterodimeric protein, right. It has both an alpha and a beta subunit. So the RGD on the fibronectin, which is present in the ECM, can now bind to integrins, which is a transmembrane protein, and that binding promotes specific kinds of cell responses. In this case, cells are able to adhere and attach to surfaces through this interaction, right. So these transmembrane proteins, they have at least two different types of interactions, one on the outside, one on the inside. On the outside, the integrins attach to or associate with, in this case, RGD found in fibronectin or any other peptide sequence found in other proteins such as laminin and so on. And on the inside, they are attached to the actin cytoskeletal filaments through the beta subunit. So this, right, and binding outside causes a conformational change that leads to a series of different biochemical pathways all the way going to the nucleus, which ultimately promotes stable cell attachment, right. On their own, these interactions are quite weak. These ligand receptor interactions are quite weak, but it turns out that cells have thousands and thousands of such ligand receptor interactions, thus making the overall interactions quite stable, right? So lots of weak interactions jointly together permit firm and adhesion to a biomaterial surface. Now shown here are two examples of those interactions. One is fibronectin, which the RGD sequence, as we said, binds to alpha 5 beta 1, which is the integrin, and this is commonly important in the cell known as a fibroblast. And another integrin, alpha 2 beta 3, binds to fibrinogen, which plays a very important role in platelet adhesion and activation. We will look at that in great detail when we look at blood material interactions in a dedicated lecture. This is just to help drive home the point that ECM interactions or ECM driven interactions are very important for many biological responses, in this case particularly cell adhesion. So they can regulate cell attachment to biomaterials through adsorbed proteins or directly through natural polymeric materials. So you can have surfaces that have fibronectin adsorbed, for example, and the integrins can recognize and attach, or if your surface itself naturally has the RGD peptide, right? Or if you have been able to graft it, and that's a very commonly used strategy, if you're able to chemically graft RGD peptides on the surface, you actually don't even need the full fibronectin. Cells can recognize the RGD peptide using specific integrins and can attach. And this integrin protein binding leads to outside in signaling. So the signals come from outside and that goes all the way to the nucleus. And so this finally results in anchoring forces to the ECM, transduction of signals from ECM to the cell, cytoskeletal rearrangement and formation of stable focal adhesions, all of which are critical for stable cell attachment. And again, this was just one example. Now, for some of you who may be more biologically inclined, I would also encourage you to look up cell migration. If you've had a reasonably good understanding of cell attachment from this slide, it will not be very difficult for you to understand cell migration, because now you want to ask, not only should the cell adhere, it has to be able to break off certain of these associations and start migrating in a specific direction, right? Cells need not always migrate in a specific direction. There's something called a random walk model that is used. But if you have a chemically imposed gradient, for example, then cells can migrate in a process known as chemotaxis. Cells can also migrate in response to gradients and mechanical properties in a process known as gyrotaxis. So the literature is very rich there. And for those of you, like I said, who are more interested in these aspects, you can look up some of the recent trends in studying cell migration on biomaterial surfaces. So now that we have a good understanding of why surface properties are important, why engineering the surface is quite important, we will now turn our attention to the engineering aspect from a biomaterial perspective for trying to modify surfaces. So we'll first start off with some general considerations for surface modification. One of the first consideration is it should be very thin and it should largely be restricted to the surface. It should not have bulk effects. So this is something you should always consider when you are looking at surface properties and surface treatments or surface modification. The idea is to ensure that you are affecting only the surface and not the bulk. And because these are surfaces, you can always have the risk of delamination. So your surface modification, whatever you're doing, should be resistant to delamination. And characterization becomes super important, not just after you have made it, but right before deployment. Because remember, surfaces can dynamically change over time, depending on storage conditions, depending on aging. This can all happen very quickly. Depending on the microenvironment that they are stored in, all of these things, surfaces can continuously change. So it's very, very important that you're characterizing and analyzing surfaces constantly. But very important to keep in mind that the surface analysis method itself should not cause large changes to the surface. So there's some tricky aspects to keep in mind. And like I said, surface rearrangement can happen with time, which can affect long-term stability. And the other thing is, surface modification can broadly be divided into two categories. One is what's done in research labs. Some of them may have no practical application, but those may be used to study specific kinds of mechanisms. But the other aspect to think about is if you're looking at clinical applications, manufacturability becomes important. So any surface modification you do, is it scalable? Can it be used for all kinds of geometries? All of these things become important. So they're both important. The former is more important for understanding surfaces from a fundamental perspective, trying to understand biological interactions with the surface under controlled microenvironments. The latter is more important for clinical deployment. So with that quick background, let's first look at very briefly the three broad classifications for surface modification, namely physical, chemical and biological. Physical modification methods are typically easier to conduct. They're often dry and rely on external energy sources to alter the surface structure or chemistry without wet chemical reaction. So that in itself is an advantage. Examples include plasma treatment, which we will look at in some detail on the next slide, ion beam implantation and UV treatment. So primarily the advantages are this is dry and rapid, it's versatile, it's not specific to specific kinds of polymers. You can do these kinds of treatments with almost all kinds of polymeric surfaces. Most of the effects will typically be confined to the surface, and processes such as plasma treatment, for example, can modify both wettability and roughness together. One of the collateral benefits of some of these treatments could also be sterilization of the material. Of course, the limitations include poor long-term stability because these are largely physically driven, and low chemical specificity, right? Because they can be used, they're not very specific to very specific kinds of polymer chemistries. That could be a limitation depending on what your application is, right? And typically plasma treatment, for example, any changes you make, unless you follow it up with a more permanent treatment such as a chemical treatment, any effects you get will not last very long, right? Even if you take very good care to store them under very controlled conditions, the surface again tends to be dynamic and it wants to minimize its surface energy, so it can change over time. We will look at that in just a bit. The other category is the chemical method, which involves reagents and solvents, essentially wet chemistry as opposed to the physical methods. And here the goal is to introduce specific functional groups, right? So you can introduce specific kinds of functional moieties on surfaces, or you can even graft polymeric chains on the surface. Now, right away you should be able to recall the example of PEG chains being grafted that we have continuously looked at through various lectures in this course. So examples include graft polymerization, silanization, which we will look at in just a bit, and self-assembled monolayers. So these may not have great utility from a clinical application perspective, but they are extremely important. They play a very important role in fundamental studies that try and look at functionalizing surfaces with specific kinds of self-assembled monolayers to try and tease out the different factors that affect specific kinds of cell responses. The advantages here for chemical methods include much higher stability, of course, than physical methods and specificity. They're also suitable for complex geometries, and they also permit secondary functionalization. So you can have a chemical moiety grafted onto something, and you can come back and graft a biological moiety thereafter, right? So they permit secondary functionalization. The limitations, of course, include the use of reagents and chemical reagents and solvents, and there's always a risk of affecting bulk properties. With biological modification methods, one can think of this as a special case of chemical modification if you're working with biological molecules that have to be functionalized. So this one example of this could simply be RGD grafting, right? RGD is a tripeptide found in fibronectin, as we just saw, that promotes cell adhesion. So you can have RGD being grafted onto a surface to induce a specific type of response, in this case increasing cell attachment, right? So examples include peptide or protein immobilization, like I just said, or you can even have drug conjugation. Another example we have hinted at earlier, and which we will look at again later when we look at thromboresistance on biomaterials, is heparinization, essentially the grafting of heparin onto biomaterial surfaces. So the advantages of the biological modification methods include very high biological specificity. It allows direct modulation of specific biological responses, and it can be made biomimetic, right? The stability can be limited depending on what you are conjugating or how you are doing it. For example, if you are conjugating fibronectin onto a surface, then you have to make sure that the conformation is retained, or at least you have to make sure that the RGD sequences are not buried inside and are exposed for cell attachment. Of course, any sterilization that you do to the material thereafter before clinical deployment can have a negative effect on the biological molecules you have attached. So these are some caveats to keep in mind as you consider different kinds of modification methods to change biomaterial surface properties. So with that, we will quickly take one example of each and look at them in some detail. So we'll first start off with the physical method, namely plasma treatment. Essentially, plasma, most of you will be aware, is dissociated gaseous microenvironment with ions, free radicals, electrons, atoms, and molecules. Now, this is typically done with air or a specific kind of a gas, again depending on what kind of functional groups you want on the surface, and it typically achieves both surface cleaning and activation through bombardment and ablation. So it removes contaminants, so it cleans the surface, and it also generates specific functional groups through activation. One very popular use of plasma is to change the hydrophilicity of the surface, right? So essentially here what happens is the reactive plasma species, namely these ions, free radicals, these unstable molecules, etc., can break the CH and CC bonds. We're talking about polymers largely, so can break the CH and CC bonds at the surface of the biomaterial, in turn forming free radicals that react with the plasma gas or ambient oxygen to introduce polar functional groups on the surface. So typically that's what is done to increase the hydrophilicity of many of these polymers that are typically hydrophobic. So one way to do that is to treat them with plasma, and for example, if you use oxygen plasma, you will likely end up with functional groups such as these on the surface. Most likely you will end up with things like these, depending on what your polymer chemistry is. Now, this is very beneficial because these polar groups can now form hydrogen bonds with water, thus lowering the water contact angle, right? Again, from the perspective of surface energy minimization, these once you have created the surface and made it hydrophilic through the introduction of these functional groups, over time these can age and the material can go back to being hydrophobic in a process known as hydrophobic recovery. Essentially, as long as it is present in air, it's not favorable for these polar groups to keep sticking out because of the increased surface energy. So the material will rearrange the surface to go back to being a hydrophobic surface. So unless you have carefully stored it under very controlled conditions post plasma treatment, the material can revert to its more hydrophobic initial state. Of course, once you deploy it in an aqueous medium, the binding with the water, the interactions with the water again cause a lowering of surface energy, thus making it favorable to for the polar groups to be exposed on the surface. And typically, while plasma treatment can be used as a standalone method, it's typically used as a pre-treatment step for other kinds of functionalization to thereafter come in. For example, later on we'll look at silanization on the next slide, and we will see that requires specific kinds of functional groups on the surface. So if your polymer doesn't have those functional groups, you can create those functional groups through plasma treatment and then follow it up with silanization. So that's typically what is done for many clinical applications. A couple of quick examples. One is we've already seen EPTFE vascular grafts earlier, where now you can, if you want to make them functionalized with heparin, what you do is on their own, they're not very reactive. It's not very easy for you to directly functionalize EPTFE surfaces. PTFE's reactivity is very poor. So gas plasma treatment creates a conducive PTFE surface. Again, it changes only the surface properties, which allows further chemical functionalization with heparin. So you see that this serves as a precursor, as a preliminary step which is now used to change the surface properties to make it conducive for further chemical modification with heparin. Likewise, you can also have catheters where insertion of catheters becomes difficult because of friction, and to ease that, what is typically done is you can treat these catheters with oxygen or argon plasma to increase the surface energy, which then allows a hydrophilic layer to come in later and permanently bond. Again, here plasma can be used as an intermediate step to increase the hydrophilicity temporarily, so that you can now have another hydrophilic polymer coating which provides a lubricious and anti-fouling surface, both for easy insertion and to prevent fouling of the material once it is inserted. Another quick manifestation of this plasma treatment is plasma polymerization, right, where you can have deposition of specific kinds of molecules on the surface. So again, depending on the surface and depending on what the monomer gases are, for example, if you have tetrafluoroethylene as the monomer gas, then the plasma breakdown in the chamber will lead to recombination on the surface to form a cross-linked film, but this will not be PTFE. It will not polymerize to form PTFE, but it will be some kind of a complex branched fluorocarbon polymer on the surface. So you can selectively now take surfaces, and if you use tetrafluoroethylene monomer in plasma treatment, you can now have specific kinds of polymerization reactions happening in situ when depositing on the surface, or directly growing from the surface, depending on what your polymeric surface is. So that is physical modification method, namely plasma treatment. We can next look at chemical treatment, namely silanization. And again, this was just one example. Silanization involves, as the category indicates, it involves a chemical reaction for modifying hydroxylated surfaces, essentially surfaces like glass, alumina, in the case of polymers, silicones, for example. And as I mentioned on the previous slide, if the material does not naturally have an OH, you can introduce the OH group by plasma pretreatment, right, to make it conducive for further silanization as the next step. The silanization technique itself is quite simple and stable because you are creating a covalent cross-linked structure. So in contrast to the physical plasma treatment method that we saw earlier. Now, silanization can typically be used to change the hydrophobicity of surfaces. An example of that is shown here, where you have a surface with the hydroxyl group sticking out, and you have this molecule coming in. And as this guy approaches under the right set of conditions, you can have methanol elimination leading to a covalent bond formation. So in that sense, it is more stable than the plasma treatment, and of course this can keep growing, right, and forming this siloxane kind of a chain and also attaching to the surface. So that's one example. So now you see here that ultimately what is sticking out here is the non-polar CH3. This will likely make the surface hydrophobic. Originally the surface had OH groups which are polar, which means the surface would be hydrophilic. But now with silanization, you can now make this surface hydrophobic, because now what's sticking out on the top are these non-polar methyl groups and not the OH groups. You can also make surfaces hydrophilic if instead of CH3 you have an NH2 sticking out at the end. So the canvas is very broad. You can have lots of designer molecules that you can use. Shown here is just one example, but depending on your application and your need, you can choose the right kind of molecule and change the properties of the surface, particularly hydrophobicity. This is typically used in the clinic in dental composites, right? We have seen dental composites in lecture 17, where we saw that it consists of a large number of components. Now there is obviously an organic matrix, a polymer matrix that undergoes curing in the presence of photoinitiated and UV light because of the methacrylate group. There are also inorganic filler silica particles that are used to strengthen the composite. Now it turns out that the interface between the silica and the organic polymer is not very stable, right? they don't naturally want to be with each other. So what happens is you can sort of couple the organic matrix and the inorganic filler through surface function of silica particles with a silane agent through silanization. So you can have 3 trimethoxysilyl propyl methacrylate, which bonds with the filler through hydrolysis of the methoxysilane groups. So this molecule has 2 sides, 2 functionalities. So on the one side, it can bond to, because of the methoxysilane groups, it can attach to the silica particles. On the other side, because of the methacrylate groups, it can undergo polymerization along with the organic matrix. So that's what we are saying here. So because of this dual functionality, now this molecule can serve as a bridge in some sense between the silica particles and the organic polymer matrix. And without the silane coupling, filler particles typically end up behaving as voids, which provide a path for crack propagation, which essentially will result in device or material failure. Silanization can also be used for catheter coatings because many of them can be silicones, these catheters. We've already seen that in lecture 17 again, the use of silicones in catheters. So you can use silanes to anchor hydrophilic polymers. You can silanize the surface can be used to now attach or graft hydrophilic polymers such as PVP to aid easier insertion into vessels, as we've just talked about in the earlier slide as well. Now the last example we're going to look at is that of biological or covalent conjugation. Now covalent conjugation from a biological moiety perspective typically requires very specific reactive functional groups such as hydroxyl or amine or acid or the SH group, particularly gold thiol interactions are quite famous, which may naturally be found in your biomaterial or can be created using plasma treatment as the pre-treatment step. Now, if the canvas for chemical modification for biomaterials is broad, the canvas for biological modification is even broader. The kinds of chemistries you can work with, the types of biological molecules you can attach are extremely large and wide, providing a large parameter space for you to look at in terms of changing surface properties, right? So we're going to look at one very famous chemistry called the EDC-NHS chemistry. I'll quickly show you the reaction scheme for you to get a sense of what kind of biological conjugation we're talking about here. But remember, this is just one of many different types of biological conjugation chemistries that can be used to graft specific kinds of biological molecules onto biomaterial surfaces. Another very commonly used association is between biotin and avidin. This is typically, it's known as the strongest non-covalent bond present in nature. It's quite extremely strong. So if you have and this is typically used in diagnostic testing, where you create ELISA plates, for example, they often exploit this strong interaction between biotin and avidin to specifically functionalize specific biological molecules onto surfaces. Anyway, so like I said, the canvas is quite broad, and here our goal is only to look at one specific chemistry, namely the EDC-NHS. So here what you have is a surface with an acid group, right? So this one could represent any surface, any biomaterial surface where you have this group sticking out. So you already have an acid group, so you met this requirement. So now we can proceed with biological conjugation. So this is the EDC molecule. This is the carbodiamide molecule essentially, the hydrochloric acid salt form. You can now use this and reaction will lead to an ester intermediate, and this ester is quite unstable, right? Now in the presence of an amine group which can come from proteins. So if you are trying to graft a protein onto a biomaterial which has the carboxylic acid group sticking out, right? So you have the group sticking out, and you can now come in with the protein which will have a free amine group at the N-terminus end, right? That can now react with this unstable ester intermediate, giving rise to the conjugated product. So you see here that you have now formed a CONH bond. So this is the biomaterial, this is the protein, right? Now EDC is rarely used on its own, again because of the instability of the ester intermediate. Essentially this can undergo hydrolysis and give you back your original carboxylic acid. So we want to prevent this, and one very common way to do this is to use it in combination with NHS. Here we are using sulfo-NHS, where now in the presence of the sulfo-NHS, the intermediate is stabilized. You form a stable ester intermediate that can again interact with or react with the peptide or the protein, the amine group, and form the conjugated product, right? So this on the top is simply just EDC. This reaction scheme is the EDC-NHS. And one example of this could be for you to graft RGD peptides onto biomaterial surfaces. So earlier, if you remember in hydrogels, in the lecture on hydrogels, we mentioned that PEG or alginate, these are all considered what we call blank slates. On their own, they don't provide or promote specific kinds of biological responses. So one way to sort of make them biologically active or responsive is to graft peptides such as RGD onto them, and some of that can be achieved through approaches such as the EDC-NHS. So with that, I hope you get a good sense of surfaces, surface properties, ECM mediated interactions, and surface modification techniques. So with that background, we are now ready to proceed to the next set of lectures that will now involve very specific kinds of biological interactions, starting from the surface, namely protein adsorption, followed by blood compatibility and the blood coagulation cascade, thrombosis, all the way up to inflammation and fibrous capsule formation.